Today's topic CRISPR-Cas9. What do you mean by CRISPR? CRISPR is clustered, regularly interspaced, short palindromic repeats. CRISPR locus is present in bacteria or archaea. When bacteriophage infects the bacteria takes the genetic material of the bacteriophage for memory called spacer, which is placed within the CRISPR region. Second time, the bacteriophage infects the bacteria, the Cas operon undergoes transcription. What do you mean by Cas operon? Cas operon is CRISPR-associated operon which undergoes transcription. The Cas operon has regulatory genes, structural genes. Where the repressor is removed, operon is turned on, creating one large polycystonic mRNA. What do you mean by polycystonic? Polycystonic is more than one or two genes which create more than two proteins. So this polycystonic one large single mRNA is not mature. Then the Cas operon has Cas9, Cas1, Cas2, etc. genes which creates Cas9 proteins, etc. proteins also. This CRISPR locus which undergoes transcription creating a long, large single mRNA which leads to short, mature single CRISPR RNA. This short CRISPR RNA contains a single specific spacer of a specific bacteriophage. What do you mean by guide RNA? Guide RNA consists of CRISPR RNA as well as trans-activating RNA. This CRISPR RNA only has the spacer. Now each short CRISPR RNA contains a single spacer, so which this spacer will go in search of the target DNA of the bacteriophage which comes for attacking the bacteria. Then what do you mean by PAM? Protospacer adjacent motif is a short DNA sequence which has 2 to 6 bases found next to the target DNA site. Cas9 protein which has two main lobes, nucleus lobe and recognition lobe. This nucleus lobe contains some histidine asparagine histidine which cuts the target DNA strand. Then it also has some RUVC domain. Then the nucleus has PAM interacting domain which helps in recognition and bind to PAM sequence. The Cas9 protein also has another lobe called recognition lobe which binds the guide RNA and the DNA. The recognition lobe types are recognition 1, 2 and it also has a bridge helix which is located within the recognition lobe, act as a connector between the recognition and the nucleus lobe which also activates Cas9. This CRISPR which act as an immune mechanism against the bacteriophage to protect its own mechanism. This CRISPR-Cas9 was discovered by two Nobel-winning scientists, Emmanuel and Jennifer. The Cas9 was first found in bacteria Streptococcus pyogenesis. After the transcription of this Cas operon and the maturation of the CRISPR RNA, then what happens? The Cas9 is loaded with guide RNA, but this only happens artificially by humans. Natural normal mechanism in bacteria where the guide RNA and the Cas9 are bind together and go in search of PAM in the target DNA. But artificially we humans load the Cas9, load the guide RNA into the Cas9 protein. Then the Cas9 scans the genome for PAM along the DNA. The Cas9 bind to the PAM first. Most of the bacteria lack PAM, which is the biggest advantage for the bacteria so that it does not cut its own genome. Then DNA unbinding begins near the PAM, not exactly near the PAM. Then the guide RNA tries to base pair with the DNA or RNA of the bacteriophage, creating DNA hybrid. Then the Cas9 activates its nucleus domain, that is histidine asparagine histidine RUVC domain. The Cas9 cuts both the strands with the guide RNA, creating double strand break in of the target DNA. The Cas9 cuts both strand with the help of guide RNA, creates a double strand break in target DNA of the bacteriophage. But one thing we have to notice is the guide RNA which tries to base pair with the DNA or RNA of the bacteriophage. This complementary strand of to the guide RNA is cutted by the histidine asparagine histidine, and the other strand of the target DNA which is not complementary to the guide RNA is cutted by RUVC domain. So this is the natural process takes place in bacteria against the bacteriophage. But if we take this mechanism in genome editing, what happens? The cell DNA repair mechanism takes place after double strand break. What are the DNA repair mechanism? Non-homogeneous end joint or a homogeneous direct repair mechanism to fix the break, enabling genome editing. Thank you.